All right, we were on the hour. You guys ready to get started?
Let's do it.
All right, awesome. Well, welcome everybody. My name is Stephanie. I'm with NEPRIS, and I just want to welcome you guys to today. Stop sharing my screen so you guys can see us a little bit better. And when you guys have questions through here, we've got some awesome presenters here for you. So be sure to make this interactive. So at the bottom of your screen, you're just going to see a Q&A button. So go ahead and type in your questions there or answer any questions that you may be asked. And we'll make sure that Owen and Karina receive them. But without further ado, I'll pass it on to you two.
Great, thanks, Stephanie. And what I'm going to do is actually utilize Stephanie's skill set here, and I'm going to ask her to read some of the answers while I'm droning on. That way we can keep track of information you're sending us, and then again what I'm going forward. So I work at OHSU. I'm the professor and chair of the Biomedical Engineering department there, and so I'm going to start probably the first half of this, just kind of giving an overview of my path toward my job, and then some of what engineers might do in the workplace in a medical school setting. And then I'm going to turn this over to Karina. She's an assistant professor, a recent hire into the department that works in regenerative medicine, and she's going to really deliver some of that really cutting edge research, and we'll finish up in that manner. So I'm going to now share my screen in theory. Here we go. Going over to Citrix. And I'm going to ask Stephanie. Stephanie, you see that okay? You're seeing something, and now I'm going to make it to a presentation. Okay, I can't hear Stephanie. Oh, perfect. Okay, now I'm in the blind, so that's why we're going to have Stephanie answer questions. So what I'm going to be talking about is an engineer's view of the vasculature. So this is looking inside a vein and inside an artery. The top part of this, that little stream of the top part, is an artery. And so blood is flowing from the right to the left. Probably flows a little smoother in life than it actually is here on our glitchy video, but we get the idea that we're having blood actually perfuse through a vessel here. The middle, those big cells, that's some structure cells. Those are very fat cells. The bottom part is a vein. That's the drainage, so all the blood coming back toward the heart. So the top part is blood pumping away from the heart. The bottom part was a vein with blood returning. And what we did here is we made a little injury. The video will start up again, and you're going to see at the beginning, yeah, nice clean little vessel. And then what happened was we made a little hole. That little zoom in, now we made a little hole. And the blood cells in the circulation are saying, oh no, there's a hole, and we need to make sure we fill that breach so the blood doesn't leak out of that vessel. And what it's doing is coordinating the activation of all these blood cells. It's making a brick wall, and then it's actually catalyzing the activation of blood components, coagulation factors, to make a mortar, to make this brick wall, to prevent blood from leaking out. So this is a classical physiological process that's making sure you don't bleed or break a blood vessel. But you see the part right there that this big blood clot just about occluded that vessel. And so what's happening is that the blood cells are coordinating, activating inside the blood vessel. And the problem is that can turn into a pathological situation where now this is how you wind up with a heart attack, or that blood clot goes and lands into the lung and you get a pulmonary embolism, or it lands in your brain and you get a stroke. And so this kind of balance between make sure your plumbing is actually not, the liquid's not leaking out, and making sure your plumbing is not making a clot is exactly what engineers study, because all of this is in the guise of transport, kinetics, thermodynamics, all engineering principles about how does biology be coordinated in the presence of all this blood flow, and then how do you actually study this to make a drug to prevent the deleterious effects of a heart attack or a stroke or a pulmonary embolism, while not allowing the vessel to leak, so not having bleeding. And so that is, the drug developers need to understand how does the engineering principles then facilitate these pathways And are there safe and efficacious druggable targets in this? So what I'm going to do is now advance to the next slide here, and I'll tell you a story where I came from. It's going to culminate over there as a spoiler alert in terms of the Oregon. You do see the little slice of the beavers where Portland is, and then a bunch of the ducks. So it depends where you guys land. But in the meantime, can all our participants, since I can't see anybody, I'm working in the blind, can the participants just send a note to Stephanie of where you're from in Oregon or South Washington there, and then what grade or what level are you? Are you high school or grade school, or are you faculty, professor, president of a university? I would imagine most of you are in high school, but why don't you send that information over to Stephanie? Stephanie's gonna butt in and start telling me while I'm telling you my story. Stephanie's gonna start telling us where our participants are from.
So we got our first one. It's Portland, and they're a freshman.
Oh yeah, all right, cool. So
another Portland from 10th grade.
Oh, perfect. So we got some Portland people. Anybody outside of Portland? So what I am, I'm nice. So I'm definitely outside of Portland. You can see that little star that's over there, over on the right-hand side here. That's in upstate New York. So I grew up in Rochester, New York. And my dad worked for Kodak, which is a chemical engineering company that used to make something called film, which people used to put in something called a camera, and that doesn't exist anymore. It does in some niche environments. People still take pictures. But in general, I grew up in that area as in the home of a chemical engineer. And then after that, I went
typing in from McMinnville.
Oh yeah, great. Well, the southern people, wonderful. And then after growing up in upstate New York, I moved down to Baltimore, Maryland, and I went to Johns Hopkins University for graduate school and worked in a chemical engineering department there. And that's where I started studying those blood cells. I spent time studying how tumor cells and how blood cells would interact while blood was circulating in a patient in terms of how cancer cells would then spread. And after that, I jumped over the coast and I went to England and I worked at Oxford in pharmacology. So armed as an engineer, I then went and learned some of the skills as a pharmacologist would to study how do actually blood cells communicate in and of themselves, know thyself, and then how do they communicate to others. So I spent a couple of years in England, and then I wandered over here to Oregon, mainly based upon the fact that I hiked the Pacific Crest Trail once upon a time, and I liked your state quite a bit. And so that really drew my eye to the West Coast and Oregon in particular, and I moved to OHSU in 2005. That's where I wound up. And so my background was in certainly in chemical engineering. And so I spent my undergrad and I spent my graduate school learning about the transport phenomenon that would be happening here, a bunch of oil distilleries, gas distilleries. How do you actually take something and then actually make a product out of it? So you have liquids, you have gases, you have triple points, and trying to actually make a column, kinetics, thermodynamics, and reaction dynamics to actually make a product. So I spent my time doing that in undergraduate. Similar, the school I went to was very similar to Oregon State, where you spent a lot of time on this left-hand picture here, deriving unit operations and actually how would you actually go about at a job making these. This looks like those kind of columns if you drive up near Albany, New York. When you look off the side of I-5 there, you see columns very similar to this. And that was what I spent my time as an engineer learning all the principles of how reaction kinetics happen and how do you actually make products out of it. But I just had the good fortune as an undergrad. I'd show you in real life, but I can't see my video right now. I've always had good veins, and so I was in an undergraduate class, and one of the students, the graduate students, asked me to give blood, and I thought that was an interesting question to be asked in an engineering class. But I said sure, and I gave blood. And what they were doing is building microscopes to study that initial movie I showed you with the blood clot. They were building microscopes to actually study blood clotting. And so I started looking as an undergrad and then on grad school and then my fellowship and now all the time as an engineer in a medical school. I started looking at the concept of the blood circulation, and then what are the principles here? Because really, this is just a chemical engineering situation. We have a pump, we have some tubes, we have some kinetics, some thermodynamics, we have some product being made, and then this is all working under normal physiological conditions. And if you make a little blood clot in that heart, in that lung, if you break a vessel in the brain, now you're going to have heart attack, embolism, stroke. And so really the balance between physiology and pathology really recapitulates what we saw from all those processes about what does oil and plastics do. And so I've spent my time looking at human vasculature as would an engineer. I see these as nodes, points of transport, points of physiology, and then druggable targets to reduce inflammation, reduce blood clots, and then when new challenges, certainly you've seen in the news with the cove, like people are throwing blood clots. You have that setting of inflammation, viral infection that is developing blood clots. So when you have a new agonist in this setting, how do you prevent those blood clots from forming? That's what an engineer in a medical setting does. And so here I'm just now going back to the actual components of the system, and I'll just tell you a couple of vignettes of the cells we study, and then I'm going to show you some people that work on these. So here's the setting. That's a blood vessel. And those are a little cartoon of things inside there. So first is you have red blood cells. You have about 4 to 5 million per microliter. So you have a ton of red blood cells, and that's the majority of the blood that is composed of red blood cells. Does anyone know, can anyone here, I can't see you guys, but maybe we have any Trekkies in here. So our blood is a little bit red and a little red in hue to it. And if you do know why, then you can send Stephanie a little note about that. And in particular, does anyone know what the color of Spock's blood or an octopus, they both have the same color of blood. If you know what the color of Spock from Trek or an octopus blood is, you send Stephanie that note, and she's going to tell us if anybody knows that. And if you do know the color of Spock's blood, then I'd like you to tell me, why is Spock's blood that color? Then we really have a real
Got our first answer. So they said blue.
Oh, it is blue. Yes, this is fantastic. Okay, so this is, was it because of the octopus, or did they know it was actually Spock? That's awesome. Yes, Spock's blood is a bluish green. Does anybody know why Spock's blood, or in our world, octopus, horseshoe crab, shrimp, any of the crustaceans, why their blood is a bluish green?
It's blue due to the copper, unlike human have with iron.
Oh, wait, did someone say copper?
Yep.
Are you guys using the Google, or you just know that? That's amazing. This is the first time in 10 years somebody actually knew the answer. It's amazing. Yeah, so the iron in the red blood cells, so the red blood cells, they're circulating millions of these per microliter, and you think you have about 3 liters of blood per person, and the iron is just a big trap. It just traps oxygen in there, and it transports it from your lungs, and then it zips it out to some tissue and releases it there. And that's pretty efficient. Inefficient are the first species to actually go about transporting oxygen, were the crustaceans and octopus and these things, and they transport it by copper. That's a pretty crummy transporter, but it's better than nothing. And that's what Spock uses, why it's blue color. Okay, so then we have white blood cells. So that's the next component. You can see how much diminished of a number. So we have 10,000 per microliter compared to millions per microliter. But the white blood cells, oh, that kind of zips off the top of the screen, but that's okay, I'll tell you. So the white blood cells are your T cells, your immune cells, your neutrophils, your macrophages, monocytes. All of these cells are kind of both on two sides. They're garbage collectors to make sure if they see something they haven't seen before, like bacteria, they eat it up. And the second part is the memory of your immune system. So people that are getting right now the Pfizer and Moderna vaccine, that what happens is your memory cells, your B cells and your T cells take a snippet of this and then go hide out in your blood, and then in comes that second dose and you make a massive expansion, and your body creates all these clones to the spike protein of the COV, and then this is what hides out in your immune system forever, hopefully, until the next time you see that spike protein, then your immune system mounts a massive response. And so your immune system has the memory part of it, and then also just has the neutrophils. It's just anytime it sees something it doesn't like, it just goes and tries to eat it up. That's why like pus is just neutrophils. That's just white blood cells trying to eat stuff up. Then the other blood cells. Oh yeah.
What is it like to do research in a lab?
Oh yeah. Okay. What's it like to do research in a lab? So in a lab setting, we want to go into here and figure out what is known and unknown. So for instance, we have those white blood cells, and you think to yourself, well, everything seems pretty coordinated. What about in an autoimmune disease? What about in multiple sclerosis? One of those white blood cells that should be just going around looking for COVID, instead found the actual coding of a neuron and starts fighting that. So you want to make a reagent, and you want to either study how that happens, or can we actually find a drug that prevents it? And so as a researcher, you think to yourself, okay, I'm going to go into work. I'm either going to purify out some cells and make a model of this. I'm going to actually study some, for instance, a microscope, and actually how do I design and study the actual interaction between a cell and a protein, or I'm going to design a clinical trial to assess patient response. So there's all types of scales that a researcher can do. You put on either you're actually pipetting out cells, you're working with patients, you're doing clinical trials. And so all of those, when a lab can take form in any of these. There's also people that do just computational modeling, like you would Amazon models what the consumers want. And so the same thing is, you can model all the blood cell responses and then make predictions of how your new drug or therapy would work there. And so a researcher in a laboratory can work on different length scales, different time scales, with blood cells, with animals, with humans, and it all depends on personally what you like, what your ethics are geared toward, whether you like animal research or not, whether the pace of it, you want to do a 3-year timeline for a patient experience and a patient response, or do you want to do something that happens in seconds and minutes? And so all of those, when it comes to a laboratory, all of those are things to experiences. I think internships and just a personal look is important for you to figure out what it personally is to work in a lab. That's a long response. So the blood cells down at the bottom here are these things called platelets, and platelets are these little band-aids. We'll look at those in a little video. And so we have hundreds of thousands of platelets that are circulating, looking for holes, and then making little patches in there. And the last component of blood cells are coagulation factors, and that's kind of like mortar. They got to make glue things together. And so if you're going to make a little hole because of some injury, or you're going to have a little gap in the blood vessel, the platelets make the little band-aids, and then the brick wall is built with these coagulation factors. Does anyone know what that mouse is there? Yeah. Yeah.
Sorry, you have a follow-up to the lab question. Someone says, I really want to research at a lab with stem cells and tissue regeneration, but I'm not sure if I can because I'm in high school.
Yeah, so Karina's talk, the second talk, is going to talk right about tissue in regenerative medicine for sure. So I'll tell you from my experience, I've had a number of high school students all the way. I actually had a freshman in high school work with me all the way for 3 years during in research and working right with blood cells. And so there's certainly lots of research opportunities to work in whatever interest area. In stem cells is interesting is there's all sorts of components of how do stem cells have some memory, how can you program them, how does heterogeneity between, are all stem cells created equal, or are there different types of stem cells in your body? How can you shift one to the other? And each of those have a different skill set that can be applied as a researcher. I'm going to leave a lot of that over to Karina for my later answer. But certainly that I've not found any issue with having high school students in my lab so far. Now, the in-person aspect of this has changed this year, as we are experiencing, because I'm yelling at you through my computer. So I've been doing some internships remotely, but that is certainly less of an experience than it would be pipetting around. So at present, I'm not allowed to have anybody in my lab from outside until at least June of this year. So there's an acute issue with my students in my lab at present. But in general, I've not had any problems with high school students. Yeah, so here I'm just showing you that one cell that is a platelet. This is the cell that makes these little band-aids. You can see that cell underneath. These are movies I did when I was in England, and I actually was the first person to use this type of microscope to actually study how a cell changes its shape. And so by doing that, that's an engineering style of approach, is I built a microscope to actually study how cells, let me move this up here, how cells would actually change their shape. And since I measured something for the first time, then I could use that measurement technique to then decipher that top cartoons of, well, how did it communicate, and what was all the cell biology behind it. So the cell biology, the components were known before, but how they were orchestrated was not known. And that's because no one had a technique to actually measure how the cells would actually change their shape. And so I made a new engineering approach, make a new tool, and that allowed me to study and unravel that mechanism up there. So that's one approach to a research. Here's the next approach, and this is actually Ishan down here. He worked with me when he was a freshman. He went to undergrad at Oregon State University, and he was in the chemical engineering department, and he worked with me as a freshman. He built this really simple model here, and it's just how blood flow and how blood creates a clot in a little glass tube. And we use this to identify a druggable target that actually years later we've taken to clinical trials and shown that this drug may prevent blood clotting during dialysis. But really, we started with this as an undergrad project, and then he went on and won a Goldwater Fellowship at Oregon State. He went to medical school at OHSU. He did his residency in internal medicine at OHSU, and now he's a fellow at University of New Mexico, working in hematology oncology. And so this is how an engineer built a very, very simple tool, studied a blood cell component that has a clinical output that really launched Ishan's career as a physician scientist. This is the next kind of component that is, we showed you originally how that model of the blood cells, the artery and the vein, were kind of coordinating that blood flow. And here I wanted to do something just on a microscope setting. How do I actually study that? Because that previous one was in an animal. And sometimes, personally, I don't like animal research. My group in general, we have 20 people. We work with animals to take things before we take it to clinical trials. But personally, I don't like it. So instead, I built these components here. I made a fake vessel. I actually used all the principles as an engineer. How do we develop a tube? How do we develop a matrix? How do we develop an imaging techniques? How do we put all that blood in there? How do we make an injury on a microscope slide? So this gave us an insight into actually, instead of having to ask an animal to do that experiment, I can now do all this work on my microscope in my laboratory. So sometimes engineering principles can be used both to figure out mechanism, as well as how do you replace some of the models to a model that you're designing in your laboratory that recapitulates some of the physiology or pathology that you'll see in animals or eventually in humans. So that's an example, again, of use of engineering principles. This is work, and I'm going to highlight another person from Oregon here. That's Michelle Burney. And when Michelle was in our laboratory, she developed a technique that actually would image the blood flow. That top image there is all the blood cells that are in blood vessels in a brain. And so down at the bottom is the normal brain. And then during a stroke, you can see all those blood vessels, the blood goes away. So we actually developed a technique when she was a PhD student in my group. How do you actually measure the effect of a stroke? And we used that to really launch a clinical trial of a drug we were developing. Once we understood a measurement technique, we can measure something, now we can study it. And once you study it, you can test a drug in it. And so Michelle used that during her PhD. She grew up in Astoria, Oregon. She went to Oregon State for her undergrad. She did a PhD in my lab. And then she did a fellowship at Harvard to understand how do you actually take this to a clinical trial. And then she moved to Washington, D.C., where she works at the National Cancer Institute as part of the NIH, where Fauci works at NIH, for instance, in terms of that acronym. And so Michelle works as one of the people there at the NIH in Washington, D.C. So again, an engineering approach, measurement technique, study biology, translated to utility in a patient. And it's the last part I'll show you here, is that actually when you do these style of clinical trials, you start with an idea and then you actually go and translate this to utility. And so what we did here is we actually get funding from big pharmaceutical companies, from the National Institute of Health, and we made a company right here in Portland. So you create some jobs and you try to actually develop techniques. Again, using engineering principles to then create product. And this is the trial that we just recently completed. So, and the last part of this before I turn it over to Karina, these are the people that work in my laboratory. Some of them raised as physicists, some of them raised with a background in statistics, many of them raised in chemical engineering, electrical engineering, and then the other half are either medical doctors, cell biologists, microbiologists. And so you mix and match in the medical field, a mix and match between how do you make tools, and then the second part is how do you use those tools to study pathology, and then how do you translate to the clinic. And I think a good example of this is actually each person that are in that picture there has a particular skill set: an electrical engineer, a chemist, a physiologist, a medical doctor. And together you make an amazing team that can actually now, again, translate that and make a difference in terms of drug development. And that's where, by choosing an engineering approach in a medical community, you really become a niche as part of these teams. So with that, there's a little picture of Oregon. I'm gonna quit my my thing here and go back to my video. Yeah, there we go. Great. Okay, and I can see, I'm going to stop sharing it there. Okay, so now we're back. We've taken a half hour for that. While Karina loads up her slides, I can actually see your Q and, oh, I can see the chat room. Yeah, great. Okay, so if there are any questions for me, I'm going to tee up Karina to introduce herself. And in the meantime, feel free to type any questions in there. So I'll turn this over to Karina.
Okay, can you hear me okay? Do you see the full screen of the slides, or do you see the like half presentation view?
It's a half presentation.
It always happens. Okay. Is that better?
There you go. Perfect.
All right. Awesome. Yeah. Any questions before we kind of jump into the next half? No questions? All right. But I'm going to just kind of get started. So Owen gave a brief introduction. I am Karina Nakayama. I'm an assistant professor in biomedical engineering. I started at OHSU about this time last year. So I had about 1 month of normalcy before COVID hit. But that was enough to kind of at least get to know the area a little bit. And I've slowly been getting to know OHSU as well. But what I thought I would do here is just kind of give a little bit of background about, my own like research journey and kind of the diversity of research that I've done throughout my career, and then what we currently are working on in the lab. And as Owen kind of alluded to, we are pretty classical tissue engineering, a bunch of different tissues that range from skeletal muscle and bone to cardiovascular like heart tissues. Previously I worked on engineering kidney tissues as well, so kind of a full spectrum using stem cells and all kinds of primary cells as well. And so with that, so my story begins on a farm. I was primarily raised by my grandparents, who were both farmers. This is in Southern California. It's in a very rural part of Southern California. I know a lot of people think that it's just like Hollywood or Bay Area with Google, but there are actually some pretty rural parts. So these are our neighbors, the goat family. And it was quite a wonderful childhood. But the main reason why I thought I would start out with this is because of my grandmother. So she was pretty much like my everything growing up. But unfortunately, she ended up getting Alzheimer's when I was probably around 10 years old. And so that is what really prompted my scientific journey. And so starting in high school and kind of going back to that question with the high school interns, I, as a high school student, reached out to a somewhat local university that was about like 45 minutes of drive out. And I was like, I want to work on stem cells. And at the time, this is a long time ago, this is decades ago now, it was brand new. Not a lot of people were working on stem cells yet. And I wanted to look at neural stem cells because Alzheimer's is a neurodegenerative disease. And so that was kind of what started my journey. I did an internship, one of very few high school students at the time who was doing internships. So I'm so glad that now it's become a little bit more of the norm. And yeah, started looking at how stem cells can be used to regenerate neurons, and thought that that was just the absolute coolest thing ever. And so then that led me to find a major in applying to undergrad that was going to allow me to continue that science. And so that was bioengineering. And I ended up at UC San Diego. So I majored in bioengineering, biotechnology there, and I also, as well, theater and dance. And so that's kind of what kept me sane throughout all of it. But it was a really fun experience. I worked a lot on like microfluidics and like lab on a chip kind of stuff. I worked with yeast cells, which is actually very different from what I ended up going towards for my grad school. But it gave a really neat foundation of what goes on that like systems level and different kinds of microorganisms. And I actually worked for a company called Invitrogen for a little while, and that has now, I'm sure that most of you haven't heard of them because they, just like all the other small companies, actually they're a pretty big company, have gotten absorbed by the gigantic Thermo Fisher Scientific that now we get all of our lab products from. And after UC San Diego, I went to UC Davis, so Californian, just kind of moving my way up the coast, where I also majored in biomedical engineering. But here I worked on engineering tissues for kidney regeneration. I worked at a primate center, and so we were looking at how to regenerate non-human primate kidney tissues. And so this is one of my images that I have up here. So this is what your kidney looks like when it's at a very high magnification. So this is called a glomerulus. That's the main like filtration unit that all of your blood will filter through, cleans it out, removes toxins, all that great stuff. And so these were some of the structures that are on like that microscopic scale that we try to make but in a Petri dish in lab. So I was completely enamored with the idea of being able to engineer tissues. I saw that picture from, I think, Anthony Atala's group where there was like this ear that was attached to a mouse. And I was like, oh my gosh, like this is the future. We're going to start growing body parts on mice. So that didn't end up actually being the future, but it was still really exciting to kind of see the possibility. So I think I'm on that like mad scientist kind of route where I think those kinds of things are cool. So not everybody's taste is along those lines. So I completely understand, but we will be talking a lot about tissue engineering in like the next 10 minutes. And so, yeah, so in this case, I worked a lot on kidneys and combined some stem cells, and we're driving them to become fully, well, a couple of different cell types that are commonly found in the kidney and getting just some very, very minor like filtration function from them. Kidney was really complicated. There's 28 different cell types. It's a lot to keep track of. And ultimately I decided I kind of wanted to change direction for my postdoc. So after I got my PhD, I ended up over at Stanford, and I decided to actually, instead of sticking with biomedical engineering, I went into cardiothoracic surgery. And I spent the next 7 years over at Stanford. And so there I worked on looking at a bunch of different materials, biomaterials, for engineering two kinds of cardiovascular tissues, either heart tissue or blood vessels. And so depending upon what kind of tissue I was looking at engineering, it kind of changed what kind of material we were using. And so in this case, for engineering a heart tissue, we want something that's going to mimic heart tissue and grow heart cells really well on. And then in the case of a blood vessel, you're looking at, these tube-like conduits that are going to transport blood and not get occluded and not, form all these embolisms like Owen was talking about. And so there's a lot that kind of goes into the planning of this, a lot on the material side, and I'll get into that in a couple of slides. But most of my postdoctoral career was looking at different kinds of cardiovascular tissues, looking at what kinds of health problems there were and where there was this unmet clinical need where we can go in and create a better alternative for patients who would otherwise just need to be on transplant waiting list. And then that's kind of what brought me to OHSU and in the Department of Biomedical Engineering. So this is our beautiful building in the bottom right-hand corner. And most recently, we've been working a lot on skeletal muscle tissue engineering, and I'll give a couple of research slides from that. But with this, skeletal muscle regenerates on its own. You exercise, you hurt yourself, but ultimately your muscle grows back. It's really, really great at doing that. There's stem cells in your muscle, and that's how we kind of get stronger. That's how we build new muscle, is we kind of injure it, and it has this wonderful ability to just regenerate. However, if you damage it too much, all of those capabilities just go out the window. And so that's where tissue engineering can really come in and see like, how can we go in and restart those programs? And we can do that using materials, we can do that using cells, we can do that using growth factors, and we can do that using other like larger system approaches, as well as drugs. And so with regenerative medicine, it's very broad, as well as tissue engineering, and that's kind of at the intersection of what our lab does at OHSU. So every research project, every experiment, every paper publication starts with a health motivation. And so on the cardiovascular side, our health motivation is cardiovascular disease. This is massive. It affects 28 million individuals. There's 400,000 new cases every year. It costs like $30 billion in healthcare. And the mortality rate, so people who die from this within 5 years of being diagnosed, is 50%. So this is a really urgent healthcare need. And so this is what motivates us to want to look at how these diseases can be treated, how they can be given alternatives to whatever standard of care they're currently getting that's giving them a 50% survival rate at 5 years. So some of the diseases that we look at are, heart attack and heart disease. So what happens kind of like Owen was showing in his video, there's this blood clot that comes in, and if it goes and gets lodged in one of the big blood vessels in your heart, it's going to cause necrosis. What happens is that there's no blood flow, and without blood flow, any tissue is going to die off, and that's what happens in your heart. And what if you happen to survive it, if you're lucky, then your heart's permanently damaged. It's weak. There's this, there's tissue that's no longer there. It's been damaged. It's gone. You just get scar tissue. And if anybody remembers, like knows when they've gotten scar tissue, it doesn't do anything. It sits there. It's tight. It's really uncomfortable. And if you have that in your heart, which is supposed to pump and move a lot, that's not going to be any good, because it's not going to be able to do anything, because it's just scar tissue. And so that's where tissue engineering and regenerative medicine can play a role. And we can go in with an interventional treatment and either remove and replace that damaged area, and then return it to a state that actually functions, is functional muscle that's going to contract, going to pump blood. But getting there and that whole process of planning and figuring out what that treatment's going to be is what we do in the lab. And another disease that we kind of look at is called peripheral arterial disease. And this is also similar to what Owen was talking about, is there's kind of this accumulation of plaques, and this can happen. It can be a genetic predisposition. It can be due to lifestyle, but essentially these plaques build up in your arteries and they block the flow of blood. And if enough blood is blocked, then the same situation happens and you don't get any blood to your tissues. They will die off, and you might even need to amputate that limb. And so that can be a really severe case. And actually a lot of people have it, especially individuals who are in the senior population, so like 65 years and older. About 20% of those people have this condition and are at risk of losing their limbs. So what we can do with cardiovascular tissue engineering is kind of all these different approaches. So that we can engineer heart valves, we can engineer blood vessels, we can create these meshes where we put cells and stem cells on them as like a patch, and then we transplant that mesh patch onto that injured area that I showed earlier from like the heart attack, when there's a lot of plaque that builds up in those blood vessels that block the flow. We can go in, cut that part out, and then replace it with an open graft that's going to stay open and allow blood flow. So those are just kind of some of the applications that can be done with cardiovascular tissue engineering. And then the thought process behind how we do all of this. There's this kind of famous triad within the tissue engineering field where we always say, okay, we need one of each of these, but now it's kind of expanded out and it's become pretty fluid. But these are generally kind of where we start when we're asking a question like, okay, we want to engineer this tissue to treat this disease. How are we going to figure out what's needed for those tissues? We start off with like, okay, it's got to have some kind of physical support. So that's our scaffold. So there's a lot of different scaffolds out there that range from like scaffolds that are made from polymers, which kind of makes sense. They've got some nice structure. You can control a lot of their mechanical properties, their porosity. And then there's some like natural biomaterials, such as your collagens and elastins that are naturally found in your body. They're found in plants, they're found in animals. And then there's some other like more creative materials, such as algae and alginate. And that material is derived from seaweed. And other materials are like if you've ever had a boba drink, those materials are actually used, not quite in the same capacity, they're not sugary, but the same basis of the materials are also used in tissue engineering. And so it just depends upon what kind of tissue and properties you're looking for as to what kind of scaffold you choose. And then you decide what kind of cells you want. Do you want stem cells? Are we going to have enough of these cells? So if we have stem cells, usually we can, if they're what we call a pluripotent, where they can become a bunch of different cells based off of, being kind of an early stem cell, then we can turn them into whatever cell we want, which is really nice and convenient. And we can harvest them potentially from the same patient that needs them. So then there's no immune rejection, which is awesome. But we do have to kind of consider like, how many do we actually need? So if we want to create a whole new heart, that's a lot of cells. And the main heart cell is a cardiomyocyte, and those do not proliferate. And so it's been really tricky in the cardiovascular cardiac field to engineer enough cardiac tissue to cover a good portion of the heart. And then the last thing is signals, and that's all of the growth factors, the drugs, to kind of get your body and the cells and tell them what to do. These are secreted factors that kind of float around near the material or the constructs, and they tell the cells, you need to become this, you need to do this. And the cells listen really well. And so that's kind of the three components that we always base our kind of thought process on when engineering tissues. And then, as I kind of mentioned, it, depending upon the tissue you want, you're going to look at different cells, you're going to look at different materials. So for example, heart, it is, I don't know if you've ever seen a heart up close, but it's going to be highly organized and aligned in specific directions. And that's because you want it to be synchronous. You want your heart to pump pretty much like in uniform, in a uniform way, so that you're not just pumping one little section of your heart and then another little section of your heart. It is somewhat kind of asynchronous, but for the most part, like, it needs to be controlled, and you can't just have like little tiny sections pumping however they want. So when we look at materials, we go, okay, we need to have really aligned materials, and we need cells that are going to contract on their own, you know. And then very differently, if we're engineering bone tissue, like, that has very different mechanical properties. So something that's soft like a heart, we can't use that same material for a bone. It's just going to squish, and then you'll have those like loose spaghetti bones, and you wouldn't want something like that. So we look at very hard materials. We look at their porosity of materials, and we really try to match the material to the original property of the tissue that they came from. And so that's kind of the main strategy for, you know, when we're looking at muscle versus heart versus blood vessels. And so a little disclaimer, if you're a little squeamish, skip the next slide. I'll keep it up for a short time, but there is a picture that's going to show a graft that has been actually replacing a bad section of a carotid artery. And so that's kind of one aspect of what we do in the lab is we create these vascular grafts out of different materials. So I'm going to go to the next slide. And so these materials are made from different kinds of extracellular matrices, like the one on the bottom right, or they're made from polymers. And we do this like using electrospinning, which is like a high voltage polymer spinner. And we create these vascular grafts that try to mimic blood vessels. So I'll get off of that slide. So anybody who was squeamish and turned away, you can come back now. And so another thing that we do in the lab is we engineer muscle. And so skeletal muscle, just like heart tissue, they're both muscle, but they have very different functions. And the way that you engineer them can be quite different as well, based off of the properties that are needed. Skeletal muscle needs to contract, but it doesn't need to contract spontaneously. But what we do want is it needs to be highly organized and highly aligned. And so this is some example of some of the engineered muscle that we have in the lab. So on the bottom is kind of a cartoon of what the muscle generally looks like. There's these fibers in the muscle. So if you've kind of ever really taken a very close look at your muscle, you'll notice that it kind of comes apart. And there's a bunch of like little fibers that are kind of all connected together and held together. And it's the contraction of those individual fibers that really produces the force that allows you to move your muscles. And so what we want to do is to create something in the lab that's going to look like that and function like that. And so we start off with a biomaterial that's going to be highly aligned in parallel. And then we're going to put some muscle stem cells and some vascular cells onto that biomaterial. And that biomaterial actually tells them how to organize. And so if we have this highly aligned scaffold, they're actually going to align, and we get these really nice parallel fibers of muscle and nicely aligned blood vessels. And so that makes it really easy for when we want to go and transplant that into a muscle injury. We have a lot of it already engineered, so the body doesn't have to do as much work. And that's kind of the goal with tissue engineering when you engineer it outside, like in a petri dish, is to try and get it as close as possible to functional muscle or to that functional tissue before you transplant it. And then here, I just have an image of the muscle that we engineered in the lab. So this is before we've transplanted it, and this is using our aligned materials that I mentioned. And so we are pulsing this to contract. So we're just kind of like, it's in a suspension of like a buffer, and we're just pulsing it with a very, very mild electrical stimulation, and it's going to contract on its own because we engineered it to do that. Now, we were engineering other kinds of muscles with different materials, and this was by far the best contraction. And so that's kind of what we do in the lab, is that we'll try a bunch of different things, different materials. As I mentioned that, you know, we wanted these aligned materials. So we tried a bunch of different aligned materials and ended up deciding that this one was the best for a bunch of different reasons, but contractile properties was at the top of the list. And to give you a kind of an idea of what this actually means, this is what happens to your muscle. So this is your tibialis anterior muscle. So that's kind of on the front of your shin area, kind of off to the side. And we're kind of looking down on it from like the top, if you're looking at your foot and you just take a section of it. So that's what you're kind of seeing. And so what happens is, this is your muscle on the far left when it's healthy. But when you have an injury to it that results in more than like 20% of the muscle being damaged, it doesn't regrow, and you just get scar tissue, and there's all this empty space. And overall, like, you just don't have the same function of that muscle. But with our engineered tissue, we can go in and repair that muscle. So everything that you see in green is what we have engineered that came from our tissue engineered muscle that we grew in the lab, and then we put it into this severely injured muscle, and it regrew really nicely. And then kind of the last thing I wanted to show you, or second to the last thing I wanted to show you in lab. So part of regenerative medicine is considering like the whole body, the whole system, and considering how this eventually is going to translate to a clinical setting. And so one of the things that, if you have an injury to your muscle or you have an injury to bone or both, then these bone surgeons will go and they will recommend physical therapy. And so when we test our engineered therapeutics in the lab, we were thinking, well, that's part of the normal prescription that is given for recovery, so we should also do that as well. So in our lab, we also let the mice exercise when we do animal studies, and this is kind of a movie that we take to kind of assess what their gait pattern is like. We have 30 different metrics that we track, everything from like the paw spreading to like the angle of the swing. So it's pretty nice and sophisticated, and we can kind of see how their recovery is doing based off of, you know, the treatment that we're giving them. And then the last thing we're doing is we're engineering like the multiple tissues at the same time. And so one of the things is your body, if you have more than one tissue that's injured, which can happen quite often if there's like some kind of traumatic accident, usually like you won't just injure muscle, you might injure both muscle and bone and the vasculature and the nerves that are in there. And so it takes a little bit more of a complicated approach to engineer more complicated tissue. And so what we've been working a lot more on recently is engineering both muscle and bone with vasculature, with nerves at the same time. And so that's kind of the current work that several of my grad students are kind of working on. And we're kind of excited for some of our like preliminary findings there. And yeah, if you ever get the chance to come visit our lab in person, this is kind of what it looks like. We have our wet lab space. This is the building to the right that we are housed in, the Center for Health and Healing. And I just wanted to leave you with, you know, overall what tissue engineering and regenerative medicine wants to do is there's these dire cardiovascular health problems that affect a wide range of people. And what we want to do is to engineer different therapies, drugs, you know, tissue engineering solutions for treating these cardiovascular health problems. And so the one thing that's missing is you. So I highly recommend science. I love it. It's my bread and butter, and I hope you do too. So any questions?
That was awesome. Both of you guys had awesome presentations. I loved it. All the pictures and the videos and stuff. Students, be sure to type in either the chat or the Q&A and let us know your questions. It's your perfect opportunity to talk with them. So let us know. And don't be shy.
And I would add that, I mean, in terms of my own experience with high school students, it's never too early to start. It's worth reaching out to labs if you're interested, just finding out if they are willing to take a high school student. You know, I think a lot of, you know, faculty are really interested in trying to train that next generation of scientists. So definitely reach out if you find somebody. I mean, if you're interested in my lab, follow up with me and we can try to see. I don't know what OHSU is like right now, as Owen was saying, but you know, we can try to look to the future for something as well.
All right, we got a couple questions coming in. Did you know what you wanted to do in high school, or did college and internships change your passion?
Is that for both of us? I mean, I guess I can go. I didn't. So in high school, I think you kind of saw my story where I was like, I wanted to work on Alzheimer's. I did not end up working on Alzheimer's. So that story definitely changed a lot. And actually, I almost didn't continue with science. I was very much a dance person, and I was going to join this local dance company where everything was set up. And I just at the last minute was like, oh, okay, I'm going to go to grad school, see how it's like. And then ended up really loving it. And it wasn't until grad school that I really was like, I need to do tissue engineering, and found that passion. Up until then, I was kind of a little bit more searching for what my science passion was going to be. What about you, Owen?
Well, you can hear me okay, Stephanie? Yeah. Yeah, I grew up, my mom was a house cleaner, and my dad had served in the military for a bit. So I grew up thinking I love the mountains. So I enlisted to go to the mountaineering division of the Army. And in between the time I enlisted and when the recruiter came to meet my parents, I tore my ACL in a high school basketball game, and then had to figure out to go to college. And I applied to go to be a physical therapist because that's what I was doing. And then my dad took me to Kodak, and I met a chemical engineer. And I'll tell you the honest answer. I met this guy in the engineering department. He was doing research on some surfactant, and as he was telling me what he was doing, he kind of like looked down at his shoes and he got nervous. And I thought, oh, that's me. That's my people. So I thought, I'm going to be an engineer just like that guy, because he got nervous in a crowd. And I thought I do that all the time. So I went to school for chemical engineering, and I just been on that path ever since. But it was definitely not a planned path. I love in the sciences the creativity, the innovation, the amazing, like that little picture of the Avengers, that each person brings in their a kind of eclectic approach to this to together create some solutions that may actually benefit people. And I think that that's a really amazing job I get to go to every day.
So then someone else is asking, what's your day-to-day life in the labs, and also how would you recommend we reach out to help in the lab?
Trina?
Oh, yeah, sure. Well, I mean, my day-to-day is probably a little different from Owen's, and it's also different from the students. So, I mean, as faculty, we spend a little bit more time behind the computer or in our offices kind of guiding and training. But I would say that up until a year ago, so my day-to-day consisted of going into lab and figuring out a couple different experiments that I was simultaneously running. I would sometimes, it was going to be a biomaterial fabrication day. So I would just make a whole bunch of biomaterials, and some of them would turn out good and some of them would get scrapped. And that can be a really intensive process. It can take all day to do something like that. I would do some cell culture. So go in, and all the different cells that I was growing, they each require different kinds of media, different kinds of attention, making sure that they don't overgrow, and just kind of maintaining them. And then the actual big days for experiments, when we have animal studies, there's a lot of prep that goes into that. So that'll be an early morning, probably get in at 6 a.m., get everybody prepped, get all the tools prepped ahead of time. And then, making sure that, everything's timed really well. So it's all like a synchronized dance, leading up to some of the bigger studies, just kind of getting those materials, getting those cells ready for those transplants. But I'd say every day is really different, and that's kind of what I love about it, is that I wasn't always just doing one thing that entire day or that entire week. Every day was different. Every experiment was different. And it's exciting because you get to build off of your results and, be creative and innovative with them.
And I'll just answer quick because I know we are at the hour here. I approach science. It's like having a really fancy restaurant, and in that story, are you actually the person putting out the final drizzle on there? Are you the chef coming up with the organization? Are you the person delivering the food? Are you the owner of the restaurant? Are you the marketing person? So my career has kind of moved through that, and perhaps at this point, I'm a little bit more in the ownership. And so I meet people like Karina on Help Recruiter here and provide the best atmosphere for her and her team to be successful, because she's the forefront of making the next discovery. And my job is just to make sure that the whole team and the funding and the environment is sufficient for her to be great. And so I enjoy that style of a job, is meeting great people and trying to make them allow them to do their job.
And any advice on how to contact labs and be able to see if they can do any internships or anything like that?
I mean, an email is always a good way to go, and I would always personalize it. If I was going to write to dear Stephanie, it was really interesting listening to you that in your work at Nepris. I've looked you up on LinkedIn and see that you've been there for 5 years, but with a background growing up in Michigan and in a time you spent in Florida. I really like to learn some career advice from you, because I think that my job with being in digital marketing and education would also, be value to me. And if that email, Stephanie's going to write back to me. And if you just write a blase email, I'm not going to catch her attention. So just that I met Stephanie today, I listened to her, and then I wrote her a particular email. That's really going to make her respond to me. And especially if you ask for career advice, because Stephanie's been like, hey, maybe she doesn't have the position, but maybe she knows somebody. So the same thing for us or anybody you reach out to. Take a moment, specialize in email, and then just write it out and try out that style.
Yeah, no, I definitely second that. Be specific. That a lot of faculty have websites that describe what their lab does. So make sure you go over that and that you come to the table with kind of like that you have kind of looked at their group and you have an idea of what kind of research you're interested in. Don't send a generic email. They might not respond to something like that. But if it looks like you've taken some time and that you are honing in on like that you specifically want to work with them, I think you have a good shot at getting a response.
Yeah, it's great advice. So we have one more question that they typed in. If you guys have just another few minutes. Someone's asking, do you guys work with genetic disorders?
Genetic disorders are a real clue to the balance between physiology, so what normally happens, and pathology. Because if it's fundamental for survival, that genetic disorder won't actually appear in the population because it'll be selected against. Sometimes a genetic disorder actually has an advantage. And so there's a balance between fundamental disorders. But is there a clue for why are there no disorders in a particular, like in coagulation factors, factor 10, no such thing as a disorder? Because if you had a disorder there, you would never be born. It's a fundamental part. On the other hand, factor 11, there's mutations. Either you have too much, you're at risk for a heart attack or a stroke, or you don't have enough, but then you bleed, but then you don't get a heart attack and a stroke. So maybe in there's this balance. And if we can figure out how to tune that, that's where our drug targets. So we use the genetic disorder actually as a way to uncover a mystery and say, if nature has been playing with this spectrum, that maybe we can target that as well. Whereas don't target factor 10 because nature's tried targeting that and everybody that had a mutation is dead. And so that's probably a bad drug target. And so that's what we learn from the genetic disorders when we're doing our discovery science. All right. Well, thanks, Stephanie, for organizing this, and it's been really a delight. And I thank you for all your time you've committed to people's careers and education as well. It really means a lot.
No, thank you guys. This is awesome. I love inspiring and working with students to help them make the best of their lives. So thank you so much for taking the time to meet with us. And thank you, students, for logging in. You got on the Q&A, someone also typed in thank you so much. So great presentations.
Great.
Yeah, thank you so much, everyone.
Have a great day.
Bye now.
Bye-bye.
Bye, everyone.